It's This Week in Creationism, episode number 35. I'm your host, Joel Duff, and here we take a look at the headlines in creationism from the past week. For episode 35, we're just going to look at one article and one particular creationist organization. It's an organization I've not talked about on This Week in Creationism before. In fact, I generally ignore this sort of branch of creationism, and that is intelligent design, or the Discovery Institute, which is often one of the biggest promoters of this idea of intelligent design. And what I want to address today is I want to give you an example of one of the reasons why I don't talk about intelligent design very much. And so we're going to ask the question, why do I ignore the Discovery Institute, by looking at Casey Luskin and The Origin of Life, a series of articles that he has been exploring along with a co-author. And while we talk about that, I'll also kind of kick in some things about DNA being copied without enzymes, because that is something that is mentioned in one of their articles. And just doing a little bit of research on my own, I discovered some kind of interesting things that I want to share with you. All right, so we have intelligent design. Why do I generally ignore it? Coming up on This Week in Creationism, episode number 35. Okay, what inspired me to talk about the Discovery Institute today? I saw something on Twitter that led me to an article from Evolution News. And if you don't know what Evolution News is, and here's the front page of Evolution News today. It's a website devoted to evolution news and science today. And you'd be forgiven if you've never been to the site or don't know the Discovery Institute to think that this is a uh, evolution promoting website where they take a look at recent articles about evolutionary biology and in the area of science, and promote that particular view. But no, not at all. This is a website that is run by the Discovery Institute. The Discovery Institute is a so what is the Discovery Institute? How shall I put it? I don't want to be too long here because I don't want this to be an extra long episode. So I'll just throw it out there that we'll call it a right-leaning, semi-political think tank that's interested in promoting and supporting certain cultural views on culture and science. And in the area of science, one of the things that they talk about most is this idea of intelligent design. And intelligent design is proposed as I'll call it a hypothesis that is an alternative to Darwinian evolution. And so it is anti-evolutionary in the sense that they are promoting a different view of what causes the variation and intricate design and complexity of life on Earth. And so when you look at a site like this, Evolution News, which is a separate website that they sponsor, it sounds like they're evolutionary, but in a way they want you to read these articles, and infused in them eventually are these ideas that, well, this is a story about evolutionary biology, except that here's why it's wrong. And so I come to this site, or was directed to this site, and the reason I even clicked on the link, because usually I ignore the articles here, and I'm going to get to why that is in a few minutes. Usually I ignore this stuff, but I happened to notice the picture that was associated with the Facebook post or Twitter post that I saw, and it showed Jeremy England in the picture. And I know of Jeremy England's work, and I wondered what they were going to say about him. So I follow the link, and it goes to an article by Walter Bradley and Dr. Casey Luskin. And the article is Still Unexplained, the First Living Cell. And in that article, you'll note that this is actually a series by Bradley and Luskin on the question, did life arise by purely natural means? Now, this would be a question that the Discovery Institute would raise many times, right? They're going to ask, how did life originate? This is one of their primary emphases, is to push the discussion about evolution to the question of the origin of life. And that's kind of the gotcha question for the Discovery Institute, right? Okay, well, maybe you can explain these various things as processes that occur in evolution that causes organisms to change and adapt to their environment, but how do you explain the first life, right? How are you going to be able to do that? And do you think that that's also a natural process? And many evolutionary biologists might answer in the affirmative that there is some natural process that led to living things being formed from non-living matter. And so say, so that's why this series. Did life arise by purely natural means, in which they're going to criticize a number of different viewpoints about the origin of life. And this is the seventh part of this series, and it's modified from a book, The Comprehensive Guide to Science and Faith, exploring the ultimate questions about life and the cosmos. Here is that book that has the general editors of Dembski, Luskin, and Holden, which are some primary members of on the staff at Discovery Institute. And here they've collected a large number of articles that is the comprehensive guide to science and faith. It's like a 600 something page volume book. And many of their books are absolutely voluminous, right? It's like the more words, of course, the heftier the book, the more you feel like, oh, there's some real solid work in here. Uh, I suspect this is a rehash of lots of other works. I don't buy their books anymore. And one of the reasons I don't is because it is like reading the same thing over and over and over and over again. All you have to have done is read a few articles, a few items by them, and you'll pretty much have caught the entire gist of intelligent design. Um, so anyway, I did find it interesting that this is an outtake from this book. So like, what caught my eye? What am I here to talk about? Besides the Discovery Institute and introduce you to them in some fashion. What is going to be the big deal here? Well, I told you already, Jeremy England. I mean, that's the only reason I went to look at this article is because I thought I'm intrigued to see how they respond to Jeremy England because I think Jeremy England has some provocative, stimulating ideas about the origin of life. And I wanted to see how they'd respond to them. So here in this first sentence, in recent years, MIT physicist, and I don't believe he's actually at MIT anymore. Let me see. Got his book right here. Jeremy England has gained media attention for proposing a thermodynamic energy dissipative model of the origin of life. England's view is summarized when he famously said that the origin of the evolution of life should be as unsurprising as rocks rolling downhill. So rather than suggesting this is some extremely, extraordinarily rare accident, um, he looks at life and says, well, what do you expect? Of course, that's going to happen. And he bases that on an understanding, a particular understanding of thermodynamics, this thing called thermodynamic energy dissipation model. And he's not the first to propose something like this, but he's been one of the, I'll say, one of the most, at some point here about 10 years ago, one of the most passionate and, you know, articulate describers of this particular model of the origin of life. And I had read a little article about him somewhere, found him to be a fascinating person, and then he had this book come out in 2020. The book is Every Life is on Fire, right? Every life is on fire. All organisms are constantly using energy, right? And they're dissipating energy. And what is that kind of energy? Generally, it's heat, right? You know, we're all hot. You know, all living things generate heat, right? So every life is on fire. The subtitle here: How Thermodynamics Explains the Origin of Living Things by Jeremy England. And I read this book about 2 years ago. I'm fascinated by the question. I don't know that much about the theories. You know, what do people think about the origin of life? Are there different ideas for it? I get asked about it all the time. And often I was like, I don't know. I don't really know what the predominant theory is. I haven't done that much reading on it. But when I had read this thing by this blurb about England, I said, you know what? I think I'll read that book. I think it sounds pretty interesting. I want to see what he has to say. And let me just read let me read a little section to you because it hits right on this idea of intelligent design. Central to the discussion will be an idea. So even in the first chapter, he's going to just give out his thesis. I have called dissipative adaptation. And since then, I've read about dissipative adaptation, which essentially is a fancy way of saying that when matter gets knocked around by the patterns of its surroundings, it ends up getting stuck in shapes that look specifically suited to respond to those patterns. All right, so let me read that again. When matter gets knocked around by the pattern of its surroundings, so molecules are found in a particular environment. That environment has an influence on the ordering of those molecules. They're knocking them around, right? In chemistry, things are moving around, and depending on the energy of those things, they're influencing other systems and other molecules. So when matter gets knocked around by patterns in its surroundings, so if there are patterns in the surroundings, certain forms of molecule, certain uh, amounts of energy flowing through a system, it ends up getting stuck in shapes that look especially suited to respond to those patterns. In other words, the response of other molecules is to take on figures, well, forms and shapes that are thermodynamically stable and are able to dissipate energy. And the whole idea of dissipative energy is that a, a molecular system that can take energy and dissipate it, which is entropy. You're increasing the disorder in the system. But if you create an ordered object that is very good at dissipating energy, overall, the whole system is dissipating energy, even if there are portions of that system that are more orderly. And it's their orderly state that allows them to dissipate energy more efficiently. Oh, what the heck is Duff Talk talking about? And what does this have to do with intelligent design? And what does this have to do with the origin of life? Well, one of the arguments about the origin of life, and just look down this paragraph here, all right? This response in this particular chapter of this book. So look at the response by Brian Miller, ID theorist, intelligent design theorist Brian Miller, to England's research. Miller points out that the kind of energy that dissipates as a result of the sun shining on the earth and other natural processes, so the sun shining on the earth is a source of energy, can't explain how living systems have both low entropy. Living systems have low entropy, meaning they're ordered systems. They have a low amount of disorder, therefore they are orderly, right? We have complex molecules compared to our environment, or complex bonds. So how can living systems have both low entropy, but also have high energy at the same time? If the second law of thermodynamics says that things are going from a high ordered state and high energy to low energy. As Miller puts it, these are unnatural circumstances. Well, I don't disagree. It's unnatural in the sense of the broad scope of the universe where things are breaking down, right? Falling apart, becoming more disordered. Energy is being dissipated, right? So it is an unnatural. Living organisms are an unnatural circumstance. Natural systems never both decrease in entropy, right? Decrease in entropy, become more orderly, and also increase in energy, not at the same time. So now this is the statement that Miller's saying. So England can't be right about this dissipative energy and thermodynamics thing, all right? Because natural systems, non-living systems, never decrease in entropy and also increase in the amount of energy they had, not at least at the same time. Living cells do this by employing complex molecular machinery and finely tuned chemical networks, right? See, there's the design aspect, to convert one form of energy from the environment into high energy molecules, right? We take sunlight's energy, not you and I, but plants do, and ultimately that energy is fed to us. And we use that energy in order to use that energy to put together and create complex molecules, right? Store energy, right? Well, at the same time, we're using energy, right? Or changing energy from one form to another. And we are dissipating energy, but that's why we have to continue bringing in energy in order to maintain our system, right? So living cells do this by taking this complex molecular machinery that's already there, and then these finely tuned chemical networks, which can be used to convert one form of energy to another and save it and store it in high energy molecules. Things that cannot be present prior to the origin of life, right? Now, here's the key. this can't have happened before there was life because they must be explained by the origin of life, right? So his assumption is the only thing on the world that can do this process of decreasing entropy and increasing storing energy at the same time, or creating complexity at the same time, are living cells. And so if only living cells can do that, how would you make a living cell if whatever came prior to a living cell is incapable of doing that? Because that's just the law, that's just the way things work. It's not possible for that to happen. So you can't get to the point of having a living cell. They freely admit once you have a living cell, you can continue to maintain that orderliness of the system and you can have complexity in a system because you have constant energy input and you have these finely tuned molecular machines that are sort of working the process and continuing to resist and buck entropy, right? As long as you are living, right? As long as your machines are working, you are able to continue to get energy from your environment and maintain all of your complexity. Now, as soon as you stop getting that energy and your systems to convert that energy stop working, of course, you're going to start to fall apart, right? You're going to decay. Entropy is going to increase, right? So the core of this argument is living things can buck the system. They're not natural, right? They're unnatural. And they're unnatural in the sense that this is not what any natural system can do. And you could almost replace natural with what a system without God could do. Whereas a system that is designed, right? That a higher being, a designer has made. And that's the thing about the Discovery Institute. They're not claiming they know who the designer is. They're just claiming that there must be a designer, right? That it is it's the end of a logic stream that's required for you to believe there is a designer because of what you see in nature and because of arguments like this, that living things can't have made themselves because the rules for making things don't work in non-living systems. All right. Without this, and I'm down near the bottom now, without this cellular machinery to harness energy from the environment and drive down entropy, right? Without that external energy to push down entropy, which it means to get rid of disorder and create order, England's energy dissipation models cannot do the task they've been handed. Right? They're just, how could they possibly work? As Miller said, England's model cannot account for the origin of biological information, which is essential for constructing DNA and parts of cells and cells themselves. All right, moving on. So he says there's something else that's really wrong here. There's a big problem, a crucial deficiency. Right? Miller has highlighted a crucial deficiency in origin of life models. That is the origin of the cellular machinery. Right? How are you going to get this machine when a machine is by definition sort of this organized thing, right? It's a complex operation and therefore has order to it. And where does order come from? There has to be something that made that order, hence the intelligent designer. All right. So how do you make that machine, much less even the simplest cell? Forming a self-replicating RNA molecule. It's a seemingly impossible task under natural Earth conditions. So it's the way the Earth operates as far as we can tell, right? It seem impossible to make a self-replicating RNA molecule, or you could just put the words there, nucleic acids. How can you make an information system that can replicate itself in a natural Earth condition? And that's still a far cry from producing all the vast machinery required by cells to exist. So he's pushing the claim back a little farther. He's saying, right, Eric, you can't make the cell, you can't even make the parts of cells, all right, with this under natural conditions. Okay, now that's where, I want to stop for a minute. And one thing first thing I'm going to say is, I've read the book by Jeremy England, and he is not claiming to be able to understand, or he's not claiming to be able to explain all of an initial cell. He's not coming up with an argument for like, here's how the cell was made, like specifically like the first cell. Here's where life began. There's lots of bold claims in here. You can't explain this. This can't be done. Under natural conditions, this is impossible. As if there was a physical law that just said, this is not possible. This is the speed of light and you can't break it. And they act like these laws are that way. They have a very simplistic understanding of the second law of thermodynamics and entropy. And that's what Jeremy England would definitely point out if he were to talk to these folks. So I read this book, Every Life on Fire, and it's about how thermodynamics explains the origin of living things. It's his hypothesis about the origin of living things. He's not really addressing directly like first cells and all the cell. He's addressing how do you go from having completely unordered or random molecules to having ordered molecules that are more complex. They have more energy stored in their bonds than the molecules that previously existed. So how do you arrange and organize molecules into complex, low entropy states? All right. In other words, more ordered. How do you create more order from disorder? And as a thermodynamics expert, he's pointing out this is inevitable. This isn't just chance or a rare event that disorder can become order in the natural world. He's not talking about in living things, right? Everybody understands that living things have the capacity to take unordered things and order them through the use of energy. Jeremy England saying there are energy sources in the world that interact with non-living molecules, and those interactions drive not unorganized molecules to become organized and become more complex. It has a whole bunch of examples, but the primary emphasis is this idea that molecules, machines, or complex apparati, right? Complex molecular bonds that are able to dissipate heat, dissipate energy, transfer electrons, move them out through the environment. Some complex molecules are able to take the energy from a system and dissipate it even more quickly in the form of heat and other things. In other words, they can assist in creating I'm sorry, I'm getting my entropy mixed up. They can increase entropy of the entire system. Even while in that system, you have complex things. And why do those complex things exist? Because they aid in the overall goal. All right, goal is the wrong word because entropy doesn't really have a goal, like there's not a thing behind it. But the property or the rule of things going from order to disorder is enhanced, or I'll say made more efficient, if you have something along the way that increases the speed or rate at which you can go from ordered to disordered. And that thing that increases the efficiency can be an ordered thing. And so molecules can organize themselves, and that's this idea of being knocked around, right? In a particular environment where these molecules are acting in a certain way and they're dissipating energy, if there are organizations of molecules that assist in the entire process of dissipation, those will naturally form, right? In fact, it's a law really that they must form. That's why he's saying it's like a rock rolling downhill. Thermodynamics tells you that there will be systems that will become more organized, more complex over time, because they are ultimately helping the goal of increased entropy. Now, I don't know if any of that made sense, okay, but because it's hard to think about entropy, and it's hard to imagine these systems. Now, my discussion here is not to get into the nitty-gritty of thermodynamics and the relative merits of different theories about the origin of life. I'm clearly not qualified to make strong judgments in this area. But I don't know if Jeremy England is exactly right in everything he says. I'm not enough of an expert to really be able to critique his work. But here's the thing. I've read enough other material, and this is where I went to this article. I saw Jeremy England being mentioned there, And I said, I wonder if they actually understand Jeremy England's arguments. Will they represent what he says? And will they actually respond to some of his more forceful arguments? I read his book, I look at this article, and my answer is no. They're just going to wipe it away with this simple brushstroke where they say, what's really important is the cell, and you can't explain the components of a cell. And anything that came before cells, anything that came before life can't work this way. Like you can't explain how order can come from disorder unless you're in a living system. And then I'm thinking, did they really read his book? Do they know that he's not really talking about the theory of how all the different mechanisms in a cell came together? Although he does have some inferences like this is what could have happened there because of my theory. He's really talking about exactly what they say can't happen, right? He's really talking about living cells do this, but natural systems cannot. Where was that? Natural systems never both decrease in entropy and increase in energy, not at the same time, right? That was the statement in there. I'm like, you didn't read this book. You didn't read this book because that's one thing he does show, right? That's what this whole book is about. That's what Jeremy England's entire idea of dissipative adaptation or dissipative thermodynamics and the origin of life. It's all about how you can take disordered atoms and why they would become ordered in a chemical system that has energy and what's the word I'm looking for? Non-equilibrium thermodynamics. If it's in a non-equilibrium state, there's going to be an attempt of molecules are going to be selected for their abilities to dissipate energy. And if the molecules that are selected for are complex because they do the job well, then those molecules are, then you're going to have an advancement in a solution from disordered molecules to some of them becoming more orderly. Not all of them, but some of them, right? Their statement that you can't decrease entropy and increase energy at the same time seems to be utterly false. Now, that's a far cry. I mean, that's a far cry from like establishing the foundational thermodynamic argument that says that, yes, complexity can increase in natural systems. And specifically, complexity of biomolecules can increase. And complexity of, say, nucleic acids can increase in natural systems apart from in living things, right? That's still a far cry from saying you can make all the molecular machines that make an entire living things, and here's what life is. Like, here's the point where life begins, right? So Jeremy England's not really accomplishing the entire task, but he is definitely he is only really interested in these initial stages of how does complexity, how does information arise. And this particular article, which talks about Jeremy England, acts as if he's a failure because he hasn't shown how cells function. But that's not really exactly the goal here. The other reason I wonder why, the other reason I think that they haven't read this book, right, is that I found the book to be not what I expected. Right? I just thought, hey, physicist, MIT, it's going to be a bunch of science. And then I start reading the book. Every Life is on Fire. Um, it's got Hebrew words at the very beginning here. And I noticed the chapter titles. Introduction. The second chapter is called Staff and Snake. The next one is Snow and Dust, River and Blood, Mountain and Sword, Flame and Tree, Wind and Breath, Voice and Word. These are all things out of the Old Testament. And for each chapter, actually every subsection, he has an excursion, a discussion of an Old Testament passage. And in that passage, actually at the end of each chapter, he goes on and talks about, in this case, the staff and the snake. right? When Moses threw the staff down and it turned into a snake. Well, what does that have to do with thermodynamics and the origin of life? Well, he's very much interested in that transition between non-living and living and asking the question, what is life? Like, what actually constitutes what makes something living? And here you have an example in the Bible of a staff, all right, non-living, although it depends on how recent you cut that stick, but because tree cells, some of them are still alive. Um, you have something that is, we'll say, non-living, and then suddenly it comes to life. And he asks really intriguing questions throughout the book about what it would take to turn that non-life into life. Like how would that happen? Now, obviously, he accepting of the fact that those are miracles because this is in the scriptures, and he is a Jew, and he takes the Old Testament to be the Word of God, and he makes that very clear in this book. Again, like I said, it was kind of surprising to read this book and kind of refreshing because it's sort of like he's looking at this and he's asking himself all kinds of questions that Christians, theists, ask themselves about the organization of life and how life came to be. And he's doing that by looking at a bunch of different really interesting and intriguing Old Testament passages like the burning bush. How does it not burn up? Well, I mean, that has to do with thermodynamics too, right? How do you stop the decay or the disorder that's occurring when you have fire, right? Every life is on fire. That tree is on fire, and yet it doesn't burn up. How could it not burn up? And so, yes, there has to be, and he kind of explains why that has to be a miracle, right? From a physics perspective. And that's kind of intriguing. But then you bring that back and you start, you go to the next chapter and you're talking about these aspects of chemistry and how certain things might be miracles, but there are other things that chemistry tells us this is absolutely what would happen. This is the design of things, of how chemistry functions. And in the design of thermodynamics and the way the normal operations of the world are set up, he begins to make an argument that naturally, although for him naturally is still in a way theistic, right? Because God is the director, right, creator of those laws. And those laws he sees as inevitably creating life, that God created matter and the way that matter interacts with each other in order to make life. Like it has to make life. That's what it would do under the right and appropriate circumstances. And then, of course, he's going to talk about those different circumstances. All right, so a really intriguing book. I really do recommend the book. Um, it's like I said, it's very thought provoking in a multitude of different ways. Okay, now back to this statement from Luskin et al. Oh, yeah, let me go here. Like the one I have highlighted here, forming a self-replicating RNA molecule, it's a seemingly impossible task under natural Earth conditions. It's still a far cry from producing all the vast machinery required for cells to exist. And I'm pretty sure England would disagree with this about a impossible task under natural Earth conditions. And I started just looking around. All right, I haven't done much reading on origins of life stuff in a while. Periodically, I'll just sort of look at the literature, see what's going on. And I wondered about some statements that Luskin had made, including this one. And so I did some quick searches, and I was wondering how many other people have followed up since Jeremy England, which England sort of suggested this dissipative thermodynamic, non-equilibrium thermodynamic origin of life idea more than 10 years ago, and along with some other scientists who are thinking along the same lines. How much has it developed since? Where has that gone? Has some of his ideas been tested? And so here's a publication from 2022 in the journal Foundations. I can't speak to the quality of this journal. In fact, my reading of the article suggests it's not the best journal in the world. I had some issues with the article, but if you want an easy reading overview of what non-equilibrium thermodynamic foundations are, and how that might equate to the origin of life, this was an intriguing article. And so he's talking about, let me just go to the thing. This particular author is making an argument about how thermodynamics actually forces you to see how molecules can become more complicated over time. And I thought this particular figure was kind of interesting. So I just pulled this out and I wanted to talk about it because I found this to be the best illustration I've seen so far of like how a molecule would become self-replicating over time. And so many people wonder, like, well, how did, if you have an original DNA molecule, like, how did it replicate? Oh, that's the other thing. Luskin actually says in a previous article, which I don't have here, but in a previous article, he says that um, you can't. I mean, you want proof that life can't have arisen by any natural mechanism. How does DNA replicate? It has to have an enzyme, DNA polymerase. DNA polymerase is this complicated protein machine that is used to grab onto the unwinding DNA and then to be able to slot in all the other bases, make the complementary strand, and therefore copy DNA. So this copying mechanism, and I teach this in class, we talk about DNA replication in bacteria and in eukaryotes, and it's a whole chapter in genetics books, and there's all these complicated proteins and different things that have to happen in order to replicate that thing. And you do get the impression from that, like, this is so complicated and such a, well, I'll use another word, irreducibly complex system, right? That how could you take any part out of this? It would just wouldn't function. And so it seems like, how would you get all those parts together at one time to do this process? And then I see this figure and I'm reading the text. And the thing here is this non-equilibrium thermodynamics, in which you have changing conditions in repeated cycles, and you have the input of energy. And when you have that, you can make complex things from simple things. And so today in class, I'm talking about PCR. Now, in PCR, polymerase chain reaction, that's a way of copying DNA in a tube, right? My students did it today. They took DNA sample. They took DNA polymerase, which is the enzyme for copying DNA. They took the parts of DNA, A's, T's, C's, and G's. So just these complex molecules, but nonetheless little bits and pieces, right? And they just put them in a tube with some other buffers, which is just all the other stuff in the solution that makes those molecules happy. And so they're in a tube, and these molecules are sitting there, right? They're not in a living thing. They're in a plastic tube. You stick that plastic tube into the PCR machine, the polymerase chain reaction machine. All that machine is a thermocycler, meaning the temperature goes up, the temperature goes down, the temperature goes up, the temperature goes down, the temperature goes up, the temperature goes down. It doesn't do anything else. That's all it does. And by changing the temperature, the molecules that are in the tube, which is an original DNA strand, just a couple strands of DNA, and little bits and pieces, which are in an unordered state, okay? Separate pieces from each other. So you get a whole bunch of little separate pieces and one original strand. And then this temperature just goes up and down. And what happens is at the end, just a couple hours later, you go from a couple copies of this piece of DNA to several million copies of that DNA. You take all the unorganized components of DNA, the individual molecules, and you build these 1,000 base pair long macromolecules, right, in a specific order. And so you go from disorder to order just by changing the temperature. Now, in that solution, to be fair, in that solution, you have nucleotides, which are the A's, the C's, the G's, and the T's. And those nucleotides are what we call charged, meaning they have two phosphates on them. They're triphosphate molecules. And triphosphates are, like, you think as little energy carriers. So each one of them is a little charged molecule. Those charged molecules then, that's the energy, not just the heat going up and down, but that's the energy so that when you add one of the A's or T's or C's or G's to a growing chain, what connects them together? You have to have energy to form that bond, to connect them together. And that's the phosphates breaking off, releasing the energy, and then you can use that energy to grow the chain. And so there's an energy form there that allows you to take simple things and make them more complex. Just like the energy from the sun, you and I are taking that energy, and it's in chemical form too by the time it gets in you, right? If you eat food that's chemical form, you're breaking those bonds, releasing the energy, and you're doing stuff with it and putting things together, right? So you can create even more complexity of other things from that energy, right? So you're resisting entropy and you're going backwards from entropy. You're creating complexity from that. Well, that happens in this tube, right? So PCR in itself, in kind of a simplistic way, is an actual demonstration that you can take molecules, simple molecules, and you can reverse entropy and you can build a complex molecule outside of a living thing, right? Now that's in a pretty controlled state, right? And somebody would say, well, you design that system in order for it to be able to do that. But you could also say, well, you could mimic natural conditions that might be able to do that. And that's where this particular image comes in. So this is a hypothesis, except that most of the pieces of the hypothesis have been shown in the lab to happen, right? And so what they're arguing here, what they're saying here is, if you go back to the early origin of the Earth, all right, you had oceans and water at very high temps, right? Most models of the early life of the Earth, it was very, very hot, all right, much hotter than it is today. And so the ocean and the ocean chemistry was at a much higher temperature, 80 degrees, 90 degrees, or something like that. Actually, 80 degrees Celsius. Um, is that right? Yeah, that's right. So very hot. However, it wasn't constant, right? You had the morning time, and then you have afternoon, and then you have night. So in the morning, the surface of this ocean is a certain temperature, or you can think of it as the surface of a lake, right? Maybe even lakes more so because they're going to change temperature a little bit more. They're not as buffered. And so the surface begins to warm up. And then when it warms up, it gets much hotter, potentially multiple degrees Celsius, right? And at that temperature, chemistry is going to act a little differently, right? Under different temperatures, chemistry, things happen differently. And then at nighttime, the temperature goes back down, right? So there's a cycle every day of the temperature rising and falling. Now, you know why I brought up PCR now, right? And so in this particular model, you have, if you have a chain of nucleotides, A, T, C's, and G's, all right? And even it's a very short chain, all right? And so you have adenine, cytosine, guanine, and thymine, which are fairly simple, small biomolecules. And it's been shown by other methods that those things can be made naturally, right, outside of living things. So it's not unexpected that those kind of molecules will be in the environment, in solution. And they will bind to each other. A's, T's, C's, and G's will have hydrogen ions bond to each other. And at about 75 to 80 degrees, they're willing to bond, just barely, but they'll hold together a little bit, right? And so you can have A, T, C's, and G's come together and form helices, or just have a single strand of them like an RNA, right? But let's talk about DNA. So let's say you have a double bonded short molecule, even if it's only 10 or 20 base pairs long, and it's just random. It's just a random piece of sequence that forms because you have warm ocean, you have molecules, and those molecules are attracted to each other, and they'll form combinations, all right? So that is one form of generating some complexity out of what seems to be simpler system. But here's where the magic comes in this case. In the afternoon, it heats up even more. And what's most important is you have UV light, a lot of UV light, especially UVC in the early atmosphere. UV light, right, will, as the water warms, the hydrogen bonds are stretched. And so the two strands of DNA are likely to come apart. Typically, it'd take a little bit warmer temperature, but with a lot of UV light, UV is energy. That energy is being caught and trapped by the DNA, right? It's absorbing that energy. And as it absorbs it, it causes the molecule to break apart. And this, again, can be shown in a lab to happen, right? And so you separate your two strands, right? So now you have two single strands. And you have lots of little pieces of these strands. And then what happens at night? At nighttime, the temperature cools. And just like in a PCR machine, when you cool the temperature down, and in the PCR machine, you have little tiny short primers that you've added in there to stimulate the process of replication, those little primers will find their matching base pairs, their complements. And so if you have billions of individual little tiny short fragments of DNA, and they're each random sequences, but even so, lots of random sequences will find their partners that share 5 or 6 or 7 base pairs. Here's a 5 or 6, 7 base pair piece of DNA, and here's another one, and they are a matching complement to each other. At lower temperatures, they will bond together, and then you will have more pieces bond. So if you had a long strand, a single strand, you'll have a whole bunch of other separate pieces bond to that. And the other great thing about this is that the ends of the nucleotides, the last nucleotide, is often charged. And how did it get charged? From the UV light. So the energy from the sun will charge that molecule, give it extra energy. And so when it binds by hydrogen bonds to the DNA strand, and there's two of them together, like, okay, this little piece matched here, this little piece matched here, this little piece matched here, this little piece matched down here on a longer strand of DNA, but they're not connected together, right? The sugar phosphate backbone isn't connected together. How do they get connected together? Well, there was definitely magnesium chloride in the environment, which is an ion, and along with the charged ends, those two allow the energy to be swapped. That energy is allowed to dissipate. Okay, you gained energy from the sun. Now entropy says you need to dissipate that. And so the way you dissipate it is you find a partner, you begin to make the hydrogen bonds, and then your charged ends, although they're stable, they're not going to release their energy. But if they get next to another molecule that they can swap bonds with and release that energy, and more energy is being released than you need to make the bond, then what have you done? You've just taken more energy and dissipated some of the energy. So you have less energy when you're done in the molecule, right? Well, that accomplishes the mission of entropy. You actually have declining amounts of energy in the system that you just ramped up with your UV light. And so you dissipated that energy, but in dissipating it, you actually gained complexity because you managed to make a double-stranded molecule as a result, and you stitched together the backbone. Now, what can happen is you can have an overlapping fragment. You can have one fragment and another one anneals to it, and another little fragment anneals to it, and they get all attached together to form the backbone, and there's a hanging end. And the next day, the next morning, they're sitting there, and then when it warms up and the UV light hits them again, they break apart, and now you have a longer strand than you had before. And then other strands anneal to it, and there's a hanging end. And the next time you can attach something to that hanging end, and you can just keep on climbing out. You keep making a longer and longer and longer DNA molecule. So you go through these cycles of warm, cooler, warm, cooler, warm, cooler, and add UV energy each day, charging the system. And so you're using energy to charge the system, and then that energy is used to make the molecules grow more complicated. And at the same time they're growing longer, they're helping to dissipate the energy in the system, right? It's really quite ingenious. And it follows from just what I did in the lab today with my students. Right? It's just applying it to molecules out in the real world that are also experiencing higher, lower temperatures and a form of energy input that can be captured by individual molecules and charging them. Now, there's another part to this, which is probably just as important, but harder for me to explain. So I'll try to give a super quick, simple explanation for. You say like, great. Okay, so you made a short DNA strand out of molecules in the environment. You constructed a nucleic acid, and you even can make a double-stranded DNA molecule, and you can even increase the size of it, right? So yes, DNA can go from simple molecules to now you have an ordered system of nucleotides. But that's just a random set of nucleotides. Like that's not like actually has information in it, right? No, okay. I don't want to go on a long side trail about what information is. But what you have is the potential for information, certainly, don't you? right? You have A, C, G, C, G, C, G, T, G, T, G, T. And although that might not mean anything, all right, floating out there as just a piece of DNA, what you're doing is you're creating potentially trillions of these little pieces, each of which have different codes. And some of those codes might actually be able to be useful for something. I mean, you might say, well, that's where the randomness comes in. Now that's where this got interesting to me again when I read this paper, is that they didn't only just say that this is a way you can create complex DNA molecules, just the molecule itself. They said, because of the UV light in the afternoon coming and striking into a lake, there's polarization of light in water. That polarization results in certain chirality, all right, certain types of molecules that are right-handed are preferred over left-handed molecules, or did I get that the other way around? Handedness is selected for. So UV light at the time of day where these molecules are going to get charged are being specific versions of molecules are being selected for out of the environment of what is probably a bunch of different molecules. And not only that, certain combinations of A, C's, T's, and G's are preferred over others because some are more stable than others. So A's and C's and G's, sorry, A's and T's and G's and C's pair differently and with different amounts of energy. And so under different conditions, where there's different temperatures, you actually select for certain sequences being more common than others. And so you might have many, many, many different things copying, but some are more efficient than others. And so you end up with particular sequences that end up being copied much more often. So entire sets of sequence that might be 20 or 30 or even 100 base pairs long that have certain patterns to them. And those patterns are the result of the amount of energy that they are dissipating and the amount of energy it requires for them to bind to each other each day. And so it's in a selective regime that is selecting for patterns of nucleotides rather than just sheer randomness. And once you have patterns, you can have consistency, right? You're consistently making certain stretches of DNA. And once you have certain stretches of DNA that are consistently made in the same fashion all the time, right, and you have a large number of them, then they can also do other things, right? DNA can bind to itself, it can fold into other structures, other proteins and other molecules can find specific sequences that are that have, if you have a consistent sequence, you can have other proteins that will be selected in the environment that attach to that particular portion of DNA and either stabilize it or possibly cause it to do something, right? That's where DNA polymerase comes in, right? You end up, what happens is you end up every time adding a little more complexity because you can make order, just because you can make DNA originally. Then you can make ordered DNA at the same time. And because you're making ordered DNA, you're actually selecting for proteins in the environment or protein-like molecules, complex molecules that are agglomerations of amino acids. And you're actually selecting for subsets of those random molecules in the environment, except you're selecting for particular ones that attach to particular sequences. And because you keep making the same sequences, you keep selecting for those particular other molecules that bind to DNA. Right, I'm not going to take the next 25 steps, but you get the idea that because you have energy in the system and just this changing in the temperature and having UV light, and the charging of the system allows an energy form to exist there that then is selected for and eventually selects for greater and greater and greater complexity over time. The environment is selecting only particular forms of molecules that are more likely to get replicated, more likely to bind to other proteins that help them get replicated, which then helps them replicate more, which then helps them become more common, which then helps them recruit other forms of molecules that make them move even better and faster. Right? So it's a feedback loop that then generates more complexity. It can only happen because there's constant input of energy, because it is true that from this thermodynamics perspective, that given no energy, systems will decay. right? Systems will fall apart. Complex things will become less complex and dissipate their energy. But given there's an energy form, Jeremy England, right? And what I'm doing right now is kind of using something of his thesis combined with some other things I've read to show that natural systems naturally will become more complex because of this form of what's called dissipative adaptation. Molecules adapt in their environment to become better dissipators of energy, more efficient dissipators of energy. And that results in greater complexity. It's really quite fascinating. Um, yeah, so just one other really quick item, and then I'm done. All right, so here's another paper. This paper published in 2020 on the thermodynamics of prediction under dissipative adaptation. So again, that word dissipative adaptation. And this idea is used in all kinds of systems now. So I just highlighted this one section, which kind of summarizes a lot of what I just said. The second law of thermodynamics plays a crucial role in the emergence of complex, self-organizing dissipative structures. So here's a thermodynamics, the second law, plays a crucial role in the emergence of complexity. Right? That statement's right there in the abstract. And so reading Casey Luskin and intelligent design folks, you make it think that that's impossible. Like outside of living systems, complex systems can't arise, right? Because they're in natural systems that have to obey the second law of thermodynamics, and so are ever decaying. And then and it's like, well, no, that's not exactly what everyone believes or says or thinks, right? That's not necessarily a law. Here we're just saying, look, the second law of thermodynamics, it isn't anti-complexity. It is a crucial role for generating complexity. It's the opposite of that. And this kind of goes along with the idea that Jeremy England and the quote that he uses about England that it's like a rock rolling downhill. This idea that you have to dissipate energy, it actually generates complexity as you're rolling downhill. As you're dissipating energy, you create complexity, and as opposed to the opposite of what most people think. I'm just reading that last sentence. This paper will argue that these two views are not contradictory. So before we were talking about two different ways to look at what's happening with energy in terms of creating complexity, but also dissipating and creating a higher entropy, all right? Reducing entropy and increasing entropy at the same time. Can that happen within one closed state? Can some of it become more disorganized while other parts become more organized? This paper will argue that these two views are not contradictory, but that their combination allows us to understand the transition from simple self-organizing dissipative structures to complex informative processing systems. Right? This paper is interesting because it's talking about information and how information can be generated or made more complex, all right, from simple self-organizing structures whose goal it is to dissipate, all right, energy, to then generating more and more and more complex systems or processing systems. Okay, not sure how to end this. I guess I get frustrated when I read evolution news, even more frustrated than when I read creation science articles. I understand creation science. I understand their worldview, what their intentions are. I know how they distort information in order to convince their audience of certain things, right? They're convinced they must be right. And so therefore, it's okay not to represent the other side exactly accurately. But here's the thing. Evolution News and Discovery Institute try to pass themselves off as not like the young Earth creationists, who even many of them think, because most of these people are not young Earth creationists. They don't believe the world is young. They believe the world is actually billions of years old. They just don't think that an evolutionary process, a natural only evolutionary process, is the explanation for the origin of life and for the diversity of living things. And so they are more sciencey minded. They think of themselves as accepting most of modern science, but they think that we've got this other angle, though, that better explains this particular area of science, right? The science of living things. And so they come across as being like better scientists, like we're more sophisticated and we have more intelligent arguments and we don't yell and shout and we're calm and we have, we have these journal articles. And so, and we have a website that looks very much like we're discussing science, right? We're not saying scientists don't know what they're talking about, well, except when you get in their literature. And so maybe it bothers me more when I read their articles and it's like, this stuff is no better in terms of understanding the literature, presenting it properly, uh, trying to put your opposition in the best light, just ignoring facts, basically just being ignorant of ideas, and then just going on with your same argument. I was like, let's repeat the same thing again. And what's the new spin today? It's just the same old argument every day and dismissive and pretending like no one else knows what they're talking about. We've got all the answers. I just don't find it's worth my time to look at answers, evolution news, or any of the stuff that Discovery Institute pumps out. And I don't find it worth my time reviewing it, talking about it. I'm only using this particular one to kind of dabble in this idea of the origin of life, and it gave me a chance to talk about this book because I've kind of thought about like mentioning it, and I wondered like how would I work this into a talk? Like how am I going to work this into something that I'm doing? And then lo and behold, Discovery Institute in Evolution News has a picture of Jeremy England and is critiquing him. And so this was my opportunity, to tell you about this fascinating book that I read. And like I said before, I don't know if Jeremy England is like, he's got it. Like, that's the answer. I just read it and I say, this is a fascinating hypothesis. And this hypothesis deserves to be tested, right? It deserves to at least be read and have you say, I hadn't thought of it that way. I haven't thought of that. Yes, maybe that's a possibility. Um, and read that from somebody who has a really fascinating perspective on things that I think is kind of a non-traditional scientific take anyway. Um, yeah. So evolution news. This is your day in the sun with me. I'm going to mention you on this week in creationism. So this week in creationism, that's it for Discovery Institute. We're done with them. Maybe something will tickle my fancy someday, but the next one, we're going back to answers in Genesis, creation science stuff. It's just more interesting to talk about and more relevant in the sense that there are a lot more people that follow young Earth creationism and fall into young Earth creationism. The Discovery Institute is a little bit a difficult animal to kind of grasp because they don't have there is a particular clientele that follows intelligent design, but they kind of touch on a lot of different fields. And there are many people I know that are like Christians in science that maybe don't know the philosophical underpinnings and so forth, and they get attracted to some of the things from the Discovery Institute because I think they do write some things that I would agree with. Uh, and so they're kind of a mixed bag of stuff. But at the end of the day, most of their intelligent design thing, even though uh, honestly, like I'm an intelligent designist, right? At heart, because I believe that God has created this world and that everything in this world is designed and created by God. And so I would like to call myself an intelligent designist. And so it feels bad having to say this intelligent design stuff is just a bunch of hokum. Um, but because there are little bits and pieces and elements in there that I appreciate, and I understand their overall sentiment, which is trying to show, like trying to find evidence in the world around us that you can point to and say, see that could only happen by, a designer being involved. And therefore, you should believe that that designer exists, right? And so I'm bringing you to the designer through the world he designed. And that's sort of an evidential argument. It's an evidence for it's an argument for the evidence, the existence of God. And I'm a tiny bit sympathetic to that, although I've never found those arguments to be particularly compelling. Um, I'm sympathetic in the sense that, yes, I would look at everything around me and say, like, yes, there's an element of design that this is designed. Now the question is, how did that design happen? And that's where I might have some differences between a person at the Discovery Institute and myself in this way. All right, so I'm rambling now, and I guess I'm being more personal. And yeah, you probably don't care about what I think. So, although maybe that's why you're here, to figure out what I think, because often I don't say what I think. I'm doing a lot of critique of what other people say, and I'm like finding errors, like, here's what's wrong, that's wrong, that's wrong, that's wrong. And I sometimes get these emails, and I sometimes get these things to say, Joel, I really like your stuff. You make some really good points. I wish you didn't make a good point there, because this is what I believe, and I think you're making a cogent argument about why I shouldn't believe that. But what you haven't done is you haven't like pointed me to, like, okay, so what is your positive, affirming, like, what is your position? Like, how should you explain this? How can you explain it? Can you make the positive argument? And often I don't do that, right? Often I am mostly just critical. And so that's something that I've been thinking a lot about. And I've been thinking about the kinds of videos that I would make that would be more of a, here's how I view the world, and here's why I think it's the correct way to view the world, and how I explain a number of different, what are my answers to a number of difficult questions about the world. So I've got a couple of those things coming up, especially stuff like from my mailbag, like, here's questions that have been asked to me. So this is a tough question. I'm going to give it a shot, try to give my answer to that question. All right. Hey, that's it. Again, my name's Joel Duff. Thanks for visiting, hanging out with me today, trying a little bit different format here because I'm at home. Um, Actually, I'm out in my wife's warehouse, book warehouse, doing this where it's quiet. And so thanks for visiting. Hit that like button and subscribe, and you'll find out what is next on This Week in Creationism and whatever else I end up talking about. Thanks a lot. We'll talk to you later. Bye bye.